it's all set. Yeah.
Okay. So I will continue in these ideas of large-scale biodiversity and connectivity. Uh, Now we'll focus more on reef fish. So we are in shallow parts than Roland showed you before. So it's easier, a little bit easier. Let's go for it. Um, so we have been for 10 years or so now working with reef fish biogeography and evolution in the Atlantic Ocean. And so we have been studying the relationships between the biogeographical areas in the Atlantic. And we now have an updated database that is showing that the three southern mid-Atlantic ridge islands are kind of getting together in this analysis, and it's nice. It's in general, it's more linked to the Western Atlantic, but you will see that some evolutionary connections with Africa is also happening. So it's very interesting place to study these isolated islands, and they are all these open ocean oceanic islands that are really different from the surrounding environment in the open ocean. And for a while, we applied general island biogeography rules that was most used in terrestrial habitats, but we are now using it for marine environments. It's nice to check richness, endemism rates, and so on the islands of the Atlantic, the oceanic islands of the Atlantic. And we now know that these very isolated and small islands have large percentage of endemic species. This is an example for fish, but it is valid for other groups as well. For instance, the St. Peter and St. Paul's archipelago. It's a tiny island from Brazil that is very isolated too. So we have some species that we have endemics that I will show later, and also some species like this queen angelfish that have different color patterns in the island. So it's very interesting what the isolation could cause to certain species. This is endemic species from there. Another one. And Ascension Island have the same idea of being very isolated and with different endemic species. So we went there and we are now working on genetics and stuff from this endemic fish, like this kind of a chocolate butterfly fish. And as one can see, they are close related to the species in the other islands. So these very isolated islands, but they have some connection, at least evolutionary connection between them. This is Chaetodon, another butterfly fish endemic. As the name Chaetodon sainte-Helene shows us. And this is more of these results from biogeography of different groups like gastropods and seaweeds, and relationships with age, area, and isolation. We now know that many species are new endemics that are relatively new species comparing to other places. So this varies worldwide, but we have more new endemics in our area here in South Atlantic. But another way to look at it is with the functional approach, instead of just going in taxonomic aspects of different species here and there, but we can also work with different traits for each species. And then we have some kind of functional biogeography to work on. And in this case, we have, for instance, calculations of functional richness in relation to isolation of the islands. So isolated islands of the Mid-Atlantic region have smaller functional richness, as we can imagine, but they have larger functional evenness, another calculation from this functional approach. So we have this warmer colorations here, close to red. And then we have also an effect of functional vulnerability. Like in Ascension and St. Helena, for example, we have many combinations of traits with just one species doing that job in the environment. So it's kind of worrying, and we have to be very careful with that. Here in Brazil, we have a large scale and long-term research program already, working with data sets, time series. We are starting from 8-9 years from now. We are working with underwater visual censuses to get biomass and density of fishes. And we also work with managers to try to establish more NPAs on those islands here in Brazil. And we have a paper published in, now it's 2 years ago, that we tried to assemble a lot of information from the different islands. And now we have some idea of species richness and comparing the Atlantic with the tropical Eastern Pacific islands, such as Galapagos here and other islands there. Density of individuals, biomass, and now we know that biomass is related to isolation. Larger biomass when the islands are more isolated, most likely because we have lower human influence. As well, we have some interaction with productivity, with area, and so on. And within the Mission Atlantic project, we have the case study SOMAR, led by Mary Gazala and me, that we will keep working, and we're going to St. Helena to explore more detailed information there. So connectivity patterns, integrated ecosystem assessment, and modeling future scenarios for these isolated islands. The idea will be to combine all these kinds of information, such as the functional traits with abundance, biomass, even interaction of fish with phylogenetic data, and then we can start to understanding more of these islands. For example, the Ophioblaenius, a small cryptobenthic fish that are spread all over the Atlantic. It's a very interesting genius to try to understand more about the evolutionary scenarios and connectivity among these islands. And it's interesting that we have when we compare with the age of the islands. For instance, St. Helena is 15 million years old, and Ascension is it's a very new island with just 1 million year old. So we are trying to understand more on these molecular clocks and the diversification of those fish. And it's interesting that sometimes, in this case here, the clay that first arrived in St. Helena arrived there more into 8 million years old, so the island was there already. But in some cases, the lineage appeared to be older than the one in Ascension Island, for instance. And they have a connectivity here from Ascension to St. Helena. So this is something to work in more detail in the near future to understand this ecological population connectivity going on. And this could happen in different clades. This one is a wrasse that's very interesting, that they have endemic species in Africa, and then in St. Helena, and then Ascension. If we put them in this chronological framework, then we have St. Helena with 15 million years old, Ascension very new. So the idea here is that the ancestral from this group came from the Gulf of Guinea to St. Helena, and then to Ascension. And it's interesting that one species, this Thalassoma St. Helena, is now found in Ascension too. So we have an endemic species from Ascension, and the species from St. Helena arrived later on in Ascension. So we are trying to understand more of all these evolutionary scenarios to couple geological information from the age of the islands and the seamounts around it to try to understand the evolution of certain groups in the islands. We have other interesting group here, some butterfly fishes like this group here probably came in the last 1 to 2 million years from the Indian Ocean. So this is interesting that from the Indian Ocean, sometimes some species enter the Atlantic, and this is very nice to understand. The Benguela current sometimes allow this passage here from linkage from South Africa and so on. So it's very nice to study these influences here. And we may have something like that in this planktivore clade too, that may be related to Indo-Pacific fish that entered the Atlantic through the Agulhas rings from Mozambique and then entered the Atlantic. Another group that we have here, another small fish, this is Cartella group. If we zoom here on the endemics for the Atlantic islands, then we have one species in Ascension, another one in St. Helena, endemics. But the separation of them is about 6 to 7 million years old. It's interesting that it's older than the age of Ascension Island. So we have all these things to think about if there is any seamount around Ascension Island that it's older then ascension, then they colonize it, then this seamount submerged, and then ascension appeared, and then it arrived there. This is something of a puzzle that we are trying to understand more in the near future. And again, this newly arrived endemics, and we have all these vagrants species going from one place to the other. We are seeing that in the last 10 years, for example, 10 to 20 years, we are seeing more and more some species going to one place to the other. So it seems that it's very dynamic, even in our ecological time frame. So we're keeping an eye on that. For instance, a lot of tropical species are arriving recently in the Canary Islands. It may be related to warming temperatures in the Canaries. So we'll keep an eye on all these distributions. So I would like to thank the Air Center for this opportunity here, and all the current funding, and Mission Atlantic, and everyone, Patricio, for the invitation. And I guess I put some ideas on what we will be working in the next few years. Thank you very much.
Thank you, Sergio, also to open up this perspective of how evolutionary biology can help in assessing the status of the ecosystem in these open ocean islands, but also more in general in different type of ecosystems. We'll come back with questions also about these human effects on these islands, but I will invite